lab we work on marine phytoplankton. And these are the organisms that use light energy. They capture that light energy and they take up inorganic carbon in the form of CO2, and they're able to make sugars and amino acids out of that and then build the components of the cell. And these organisms, because they have this amazing ability, are really at the base of marine food web. So they're incredibly important to the life of the planet. And these organisms have been studied, I'd say, for decades really. But what's really fun about science is that it keeps changing, we keep learning new things, and we keep getting new perspectives on these organisms. And one of these developments that's come about is the use of satellites in oceanography. So that basically satellites now give us a chance, because they see the chlorophyll molecules of these organisms, to map the phytoplankton over the whole globe. So instead of studying the water in one place, off say Scripps Pier or another water sample, we can actually follow the phytoplankton over the whole planet. And this is a satellite image provided by NASA showing the globe in February of 2000. And what you can see is places where the phytoplankton are very happy and blooming. For example, here, these kind of green colors. There's lots of chlorophyll in the water and lots of phytoplankton growing very rapidly. And then there are large areas of the ocean, as you know, that, as seen in these blue colors, have very few phytoplankton. So there's very little chlorophyll in the water. But not only do phytoplankton vary in space, they also vary in time. And so this is, again, provided by NASA. They're able to have a month-to-month shot of phytoplankton chlorophyll on a global scale. And you can see that basically the phytoplankton spring bloom occurs up here. This changing is growing into the green color, green-yellow color, and along some of these coastal regions. And this is just a way that summarizes that. That kind of takes the whole yearly data set and actually boils it down to one picture. And you see, again, these areas of the ocean, very few phytoplankton. But these, even though there are very few phytoplankton here, they're actually a huge part of the globe, so they're still very important in global carbon fixation. And then areas where we actually have upwelling regions where there's a lot of phytoplankton. So that's been a revolution in terms of trying to understand phytoplankton. And it really harks back to really the questions that have dominated some of this field for a long time and still are. And some of these are, what organisms are present? So even if we see chlorophyll, what phytoplankton species are there? Are they big cells? Are they little cells? What's their, how diverse are they and why are they diverse? It's been a major question in marine biology and still is. How fast are they growing? Are they growing very quickly? Is it a very active ecosystem? Or are they growing very slowly because there are very few nutrients? And then more broadly, what are the chemistry and physics going on that's affecting those growth rates of the phytoplankton? How fast are they taking up carbon and fixing energy, fixing carbon? And what biological factors are also involved? Who's eating those phytoplankton? How is that carbon getting transferred up the food web? So these are some of the basic marine questions in marine biology that dominate the field and have motivated a lot of the questions that have been asked in this field. So just as satellites started to give us a global perspective, there's a whole new technology called genomics. And genomics is giving us a perspective in the cell at a very reductionist level, each base pair in a phytoplankton. And again, providing a whole new perspective on this field. And so I wanted to kind of just go over some of the basic terminology of genomics and what some of these words mean, and then talk about how it's impacted my own research. And the first word is just, what's a genome? A genome is just another word for the blueprint of a cell. And so if we have an object like a house, which you, for example here, I just picked this off the internet. A blueprint is, as you know, the plans for that house. It tells an architect or a builder how to build that house, how to replicate it, and where the various parts of it go. And a genome is just the instructions written in the DNA for how this organism should replicate and build another copy of itself. So it's just the instructions to a cell and written in nucleotides in the form of DNA. And so here's the object that we work on, an organism called marine Synechococcus. And here's two examples of blueprints. So that house I previously showed you, here's a blueprint of that house. You can see the dining room, the kitchen, living room, whatever, and actually the toilet. And here is the blueprint of Synechococcus, marine Synechococcus, that organism I showed you a picture of. And in this representation, this is the DNA molecule in the cell, and then each one of these little wedges is a gene in that organism that it codes for a protein. And some of those proteins do the same kind of things as toilets. So cyanobacteria have what are called efflux pumps, and they pump waste out of the cell. So this is essentially two different ways of mapping and describing objects, and this is the genome of marine Synechococcus. So what's revolutionized biology in the last 10 years or so is our ability to sequence DNA and actually map a whole genome. And I just want to explain briefly how that's done, because it was a revolutionary breakthrough. Basically, in terms of technology, we can sequence 700 base pairs of DNA approximately. We can't sequence a whole huge molecule of DNA. And what Craig Ventnor and Claire Fraser and a lot of collaborators figured out is if you have a genome of an organism, and say you have multiple copies of it, if you take that DNA and break it up into lots of little pieces, and then you do this sequencing trick. You sequence in about 700 base pairs from each end, because that's about all you can do. But then it's like a jigsaw puzzle. You have these pieces, you sequence in, but you can actually piece those together and figure out the whole genome. So you can figure out a lot more than 700 base pairs. You can actually figure out millions of base pairs by puzzling them out. And so this is done routinely now. It was done with the human genome, and it's being done with marine organisms, as I'll talk to you about a little bit. And I wanted to introduce one more term, and that's called ecogenomics. And the reason for doing that is there's a paper in Science this week. It was one of the first applications of this to marine systems by Craig Ventnor. And ecogenomics just means we look at very complex samples. So here we have a seawater sample, and each one of these, imagine, is the DNA from one bacterium. And so here we have these two bacteria are the same. Here's their DNA, and here's the DNA from two others, and here's another one. And ecogenomics just means let's just take all that DNA together, we'll shear it into little pieces, sequence in from 700 base pairs on each end, and then we'll do what's called assembly. We'll try to put those together. And what's amazing is that they can start to reconstruct organisms that from their DNA in the water column. You don't need a pure isolated organism and pure DNA. You can take these very complex samples. Now they can't quite piece together all the organisms and all their DNA, but they're making this, they're actually being able to see lots of the organisms that are there and huge chunks of DNA. And we're starting to get a sense of organisms that are in the water even though we can't culture them. And that's going to be a kind of a real novel breakthrough again in marine biology. And that's just happening last week. That's kind of some of the excitement happening in this field. So that's ecogenomics. So let's get back to the organisms. This is the organisms that I'm particularly interested in. These are called cyanobacteria, and the first one is called Synechococcus. And Synechococcus is about 1 micron in size, and it was discovered in about 1978, not that long ago, when John Waterbury was looking through what's called an epifluorescence microscope, and he could see these little orange dots, and he realized that those are cyanobacteria, and so he isolated them in culture and described them, and he showed they use what's called a phycobilisome, a very particular light harvesting apparatus. And then about 12 years ago now, there's another discovery. So again, only about a decade ago, we discovered an organism called Prochlorococcus. And this is another cyanobacterium. It's actually smaller, and it uses slightly different pigments to harvest light. And we now know that this is the most abundant photosynthetic organism on the whole globe. And this was only discovered about 12 years ago. So we're still making these fundamental breakthroughs in this very simple question of who's there in the water. So both of these organisms are particularly important in those blue open ocean areas that I described. But then Synechococcus actually exists right here off our pier. It's very abundant right there. And Synechococcus has two other kind of useful and interesting aspects. Some of these strains know how to swim. They kind of zoom around, and actually we don't know how they do that at all. It's sort of, they don't have an appendage called a flagellum that's found in other swimming bacteria. So they're swimming, and we don't know how they do it, and it's a fundamental problem in microbiology. And they can swim, we think, towards still nitrogen compounds. So they get nutrients that way. They detect patches of nutrients and they swim towards them. So we kind of know why they swim, but we really don't know how. And we can, we have what's called a genetic system. We can manipulate the DNA of this organism. And I'll give a brief example in a little bit later. So there's some nice properties of Synechococcus. And here's what I call the credits. So if this were a movie, this is what would be rolling down at the end. These are the people who did all the work I'll be talking about. And I want to particularly note that the sequencing, so this was done by the Joint Genome Institute of the Department of Energy. So they did the work to really sequence those 700 base pair pieces and then put it all together. And then Patrick Chain did what's called the finishing, and he kind of, there were a few little holes in that puzzle, and he put the last little pieces in, which is very important. And then this group of people here, Frank Larimer and Miriam Land at Oak Ridge, and then my collaborator Bianca Brahamsha at SIO, and then John Waterbury, and this group of people here then looked at that DNA and stared at it for about 2 years and put together some of the puzzles I'll be telling you about. So one last word, annotation. So remember I said we had this DNA, and now we know the whole sequence of the genome of an organism, but that's just the base pairs. Those are just this string of T's and C's and A's and G's here. And annotation means you take that information and then you try to figure out where the proteins are and what those proteins might be doing. Okay? So here's this arrow here is to indicate that this is part of a protein that's been predicted to be coded by this DNA here. And this is another protein that's predicted to be coded by this DNA as well up here. And this is a blow-up of that because in the whole genome, there's lots of these segments of DNA that code for proteins. And so what we needed to do is take that DNA information, figure out where the proteins were, and then try to figure out what those proteins do, because that's what's gives us insight into the genome of Synechococcus and what it's capable of, and so that's annotation. So we spent quite a while doing that, and it turns out there's 2,526 predicted genes in this organism. We call those ORFs, so I might slip into that kind of technology speak. So there's 2,526 ORFs, or possible genes in the organism. And what we end up with is this genome map shown here, and each one of these wedges was a predicted ORF in this organism. Okay, so some of the insights that we got out of having this genome. Basically, we got a lot of insight, we thought, into the life of the organism in the oligotrophic environment. So you have to realize we take these organisms out of the water and we grow them in the lab, and we grow them under pretty unusual conditions. And so it's hard sometimes to know what they're capable of and how they're really experiencing the open ocean environment that they live in. And one of the things we saw from the genome is they have very little what's called regulatory abilities. They have very little ability to respond to changes in their environment. And that was kind of a puzzle, because we sort of thought, well, they should be able to respond really quickly, or why not? For example, E. coli, a very common bacterium, has a very complex regulatory system. It has about 40 proteins involved in sensing all kinds of things in its environment. But Synechococcus only had about five of those systems. So you can see it's a huge reduction in what we call the regulatory ability of the cell. And we think that's because these organisms have adapted to a ligotrophic environment. They haven't adapted to the lab. They're used to seeing very, very low nutrients. The conditions don't change very often. The light comes and goes on a daily cycle, but basically conditions are pretty constant, and we see that, we think, kind of inscribed in their genome. I'm really glad you got to hear Kathy Barbeau. Kathy works on iron, as you know, and iron is a very important element we're realizing in the marine environment and can control how phytoplankton grow and whether or not they're able to survive. And one of the things we see inscribed in the genome of our organism is some novel abilities and ways they've tried to get around not having enough iron. And one of these was an enzyme. Instead of using iron, they used a nickel in an enzyme called superoxide dismutase. So you probably know that there are free radicals generated all the time in biology through light and in our own bodies. And organisms need ways of getting rid of those free radicals, and they have an enzyme called superoxide dismutase. And that's often an iron enzyme. But in our organism, this organism figured out how to use nickel instead, so it doesn't need as much iron. And so we think that's, again, a reflection of this interaction of biology and chemistry going on in the environment. And there are quite a number of examples of that in which ways they saved iron. So the genome is also changing our perspective on these organisms. We like to think a lot about, is something a plant or an animal or a mineral or whatever, but phytoplankton are really different. They're evolutionarily different from plants. And even though we think of them like plants, they use light and they have that similarity, they're not plants. And this was really enforced by the genome. So these organisms, we can see in the genome that they can take up organic compounds, amino acids and other organic compounds. And that's not something you'd really predict a plant to do. Usually we think of plants as things that we fertilize in our garden and we give them phosphate and nitrate or ammonia. Inorganic compounds. But what we see in the genome is these organisms can take up a lot of organic compounds, and that was something we really didn't expect. So it's kind of changing our perspective on how we think of these things. We can't think of them as plants, we can't think of them as animals. They're really another group altogether. One thing that I was really surprised by is there are pieces in the genome of foreign DNA. So even though these organisms that live in an environment where there's very few cells and very few nutrients, they've somehow picked up DNA from other organisms. And the way we saw that, you can see in this genome map here, each one of these little regions here where it dips down, here, these places here, have a very anonymous DNA composition. We call that %G+C, but it's very anomalous. And it's actually an indication that there's foreign DNA that's somehow gotten into this organism. And what was even more interesting was this DNA contains things that actually would be useful to Synechococcus. So it kind of makes sense that it's found these foreign pieces of DNA, and they actually encode for proteins that might be very useful to the cell. And one group of these proteins are called glycosyltransferases. And what that is, it's just a word for enzymes that put sugars on proteins. And so our hypothesis is that these organisms, here's my cartoon for Synechococcus, and they're eaten all the time by a grazer. About every other day they get eaten. So they're constantly hoping they could avoid being eaten, because there's a good chance that's how they're going to die. And the other possibility is that they'll actually get attacked by a phage. Those are kind of two of their fates in life. But these enzymes, glycosyltransferases, could put sugar molecules on proteins on the outside of the cell. And what that would do, it'd be like camouflage. Essentially, if the organism, the Synechococcus, looks different, then maybe that grazer that's trying to find it in this dilute environment won't find it anymore. And it'll be able to elude the grazer for a little while longer, or maybe a phage for a little while longer. So this was a class of enzymes that seem to be transferring around in the environment, possibly acting as camouflage. So that was just kind of at the beginnings of an insight. And so it's a hypothesis, and it's something we now can go in and test, that we have the genome. There's another area of genomics called comparative genomics. So again, I said we're just starting to even now get a handle on what cyanobacteria or phytoplankton are in the environment. And I've kind of given you two groups, Synechococcus and Prochlorococcus. But really, those are names now we're understanding for a whole group of organisms. And what this is here is we now have the genome of Synechococcus, and we have the genomes of several Prochlorococcuses. And when you line them up, these are genomes lined up, and then you draw lines between any two proteins that are identical. And you kind of come up with this map. So here, for example, this region here is very, very similar to this region here, which is actually pretty similar to this region here. So even though what was this insight we're getting is that even though these organisms, for example, here are both called Prochlorococcus, you can see this one has a much smaller genome than its sister Prochlorococcus, and then our Synechococcus. And what we really don't know why yet. All we know is that this is a very different organism. It has a lot less DNA, and its DNA codes for different things than the other Prochlorococcus, and than it does for Synechococcus. So what genomics is doing for us as we get more and more is it's starting to help us understand diversity of phytoplankton in the environment and why they might be diverse. So I'm going to talk quickly about how we use genomes and genetics. And what's nice about Synechococcus, as I mentioned, is that we can manipulate its DNA and in the lab create organisms that are different than the field. So we can take out DNA and put DNA in, and that's called mutagenesis. That's the word we use for that. So we can take out a specific gene if we have a hypothesis, and we can put genes in. And this was just a quick example. Remember, I mentioned that we were really interested in the ability of these organisms to take up organic compounds like amino acids, because we didn't expect that phytoplankton would do that. We thought they were more like plants. So we tried to do that in the lab. We tried to say, okay, here we'll take Synechococcus and we'll add in some amino acids and see if it can grow on those as predicted by the genome. And so often, as happens in science, what happened is that Synechococcus died instead of growing. It's exactly the opposite of what we predicted. So here we have Synechococcus. These are cells, and these are in days. And here's Synechococcus growing happily. And here we gave it some amino acids, and they just stopped growing. So that was a bit frustrating because it was not what we predicted. And we tried to be good scientists and come up with some hypotheses. And we hypothesized that Synechococcus, since it's adapted to a very dilute environment where there's not very many nutrients, has a very minimal regulatory system, it just can't stop taking up something. So it really likes amino acids, it just can't stop, so it can't regulate very well. And we actually think this is a very general possibility, that we have a lot of trouble growing bacteria that come from these ligotrophic environments, and it might be that they're not used to very even low concentrations of things like amino acids, and they just die. So this is our hypothesis. And one way of testing that was saying, okay, let's take out the amino acid transporter and see if the cell lives. And that's actually what happened. So here we have a gene called SYNW0828. That's a protein encoded in the genome. And we, using genetics, took out that gene, we knocked it out, and we tried to see what happened to the cell. And what you can see here, these are what we call the controls. And now we've added back alanine or serine and several amino acids, and now the cells grow happily. So if we took out the transporter, so it can't transport anymore, and now they're fine. So it's not something toxic about amino acids to the cell. It's just that they don't know how to regulate their transporter. And when we did it for them, then they're fine. So it helped us confirm a hypothesis that came out of the genome. And that's one of the powers of genetics, actually. So and I just want to give you a really quick overview of kind of where this field is going, or where I see it going. Our lab has been funded to sequence another Synechococcus that lives off the pier. And the reason for doing that is it's a very different environment off the pier. As you know, it's changing all the time. It's very different from a ligotrophic environment. And so we're going to compare a pier Synechococcus to an open ocean Synechococcus to try and see how that diversity, how different they are. And another one I just wanted to mention quickly, because it lives again right off the pier, is what we think is the world's smallest phytoplankton. So this phytoplankton is called Osteococcus. It's about 1 micron, so it's the same size as Synechococcus. It's really tiny, and it coexists with Synechococcus off the pier. And actually, it's more important than Synechococcus off the pier in terms of carbon fixation. So it's very important. But we don't understand how it competes with Synechococcus, and why is it so small. And these are things that we think we'll see in the genome. So JGI has actually just finished some sequencing from this one, but we haven't even seen any of the data yet, so I can't tell you much about it, other than it's about 12 megabases. So again, it's about 5 times bigger than Synechococcus. So it's much bigger, but it's the same in its genome, but it's much the same size physically. So what I've tried to do is kind of give you a bit of a whirlwind tour, and what I see is the ways that genomics is revolutionizing marine biology. And I mentioned really briefly this term, ecogenomics. I think you'll see a lot more of that in the next five years or so. And it's going to help us understand organisms in the marine systems without culturing them. And then genomics of single cells is really taking off now. And that, as I said, is, I think, giving us a real sense of the blueprints of cells of these organisms, and it's helping us infer how they're living in the environment. And then what it's also doing is it's going to let us develop ways of probing and studying those organisms back in their environment. So we can actually use the genome information to develop tests and probes to study organisms back in the environment. And lastly, I just want to acknowledge my SIO collaborators, since you may have met some of them. They've been incredibly important in these projects. So Bianca Brahamsha is at SIO. She's involved in the Synechococcus genome project. And then Mark Hildebrand is one of the people in the Thalassocyra diatom genome project as well. And then this is paid for generously by the Department of Energy, which is really interested in these organisms because they're very interested in how carbon gets put into the atmosphere through energy, and how we might be able to get carbon out of the atmosphere using organisms. And then NSF also has a program to sequence some of these organisms. And I'd like to thank all of you for listening this morning. Thanks.